So what is mitosis? Mitosis is the process by which a single cell divides into two daughter cells, and these daughter cells should have identical genetic makeup. The main responsibility of this kind of cell division is to ensure that there is equal distribution of chromosomes into the two daughter cells, and the chromosomes in both the cells are identical. This kind of cell division occurs in non-reproductive cells, which are called somatic cells. What are these kind of cells? Like brain cells, skeletal muscle cells, or the blood cells, cells in your organs, heart, skin, all over the body. The importance of this kind of division is it leads to the growth, tissue repair, and asexual reproduction in some lower organisms. The life cycle of a cell is called the cell cycle. The cell cycle has three main phases: interphase, mitosis, and cytokinesis. Higher organisms, including human beings, have diploid genetic makeup, which means they have chromosomes in pair. Like you must have seen, we write 2n, and human beings have 23 pairs of chromosomes. 23 pairs means 46 chromosomes in total. So diploid organisms then undergo DNA replication. The replicated DNA forms a chromosome, and these chromosomes are then divided, with half of the copy going to one cell and half of the copy going to other cell. And finally, these two daughter cells separate. So in interphase, the cell prepares itself by ensuring that the DNA has properly duplicated and all other organelles are also ready for division. In mitosis, there is actual separation of these duplicated chromosomes, and in cytokinesis, the cells actually separate into two. A cell spends majority of its time in interphase. This is the phase in which a cell performs its normal function. For example, if it's a neuron, it performs the function of conducting nerve impulse, and if it's a muscle cell, it performs the function of contraction and relaxation. The interphase is further divided into three phases. G1 phase, in which the cell performs its normal functions and spends most of the time in this phase. S phase is the synthesis phase, in which DNA replication occurs and the chromosome makes its own copies. G2 phase, or the gap phase 2, is the phase in which cell prepares for mitosis by synthesizing proteins essential for the progression of cell cycle and organelles. Interphase is not a part of cell division. It is the phase in which the cell normally exists. Cell division occurs towards the last phase, when the cell has grown enough and is now ready to divide. Mitotic phase has further four phases: prophase, metaphase, anaphase, and telophase. Remember, the goal is to ensure that both the daughter cells receive identical sets of chromosomes. That is why if you take any cell from your body, it will have same genetic makeup. So if you remember the structure of a cell, what you're seeing here is a cell. It has cell membrane, centriole, endoplasmic reticulum, mitochondria, and the important player here, the nucleus. Inside nucleus, you see chromosomes, and this structure is the nuclear envelope. So in prophase, the chromatins condense to form chromosomes and they become visible. Nuclear envelope starts to disappear, it starts to dissolve, and spindle fibers are formed from your centrosome. So the key structures formed during this phase are centromeres, chromatids, and spindle. During the interphase, or most of the life of a cell, you will see chromatins inside the nucleus. The chromatin condenses to form chromosomes during the prophase, and the chromosomes have a centromere in its center. So what happens during metaphase? During metaphase, the chromosomes are aligned at the metaphase equatorial plate. So you will see that metaphase chromosomes look like this. They align in a line. By this time, the centriole has also migrated to the poles, and there are spindles arising from the microtubule organizing center of centrioles. These spindles are attached to the centromere, and they align these chromosomes in the equatorial plate. It's very important that all the chromosomes are properly attached to the spindle and they are ready to separate. Now the chromosome that you see during the prophase will appear like this. So this is a chromosome and this is a duplicated copy of that chromosome. These two strands are called sister chromatids. These sister chromatids are then pulled apart by the activity of these spindle fibers. The chromatids then move to the opposite poles in this cell, and each pole receives an identical set of chromosomes. In the telophase, the chromosomes that had reached the pole now start becoming chromatids by decondensing. Here you will see that the nuclear envelope has started to reappear, the spindles have started to dissolve, and finally you will have the beginning of two cells separating out. By now you have two nuclei per pole. Cytokinesis is the actual phase where the cytoplasm divides, which will ultimately lead to the formation of two separated daughter cells. In animal cell, you will see the formation of this furrow called the cleavage furrow. This cleavage furrow ultimately joins and two daughter cells are formed. In plant cells, what happens is small vesicles align in this area and they fuse to form the cell plate, which leads to the separation of two daughter cells. Now let's look at the significance of mitosis. Mitosis ensures that the daughter cells receive identical genetic material. It ensures that all the cells in our body have the same genetic material. This kind of division is also important for the growth of organism. It also helps in tissue repair whenever you have injury and you have some scars. This kind of division leads to the repair of that injury. In the unicellular organisms such as the paramecium that you're seeing here, mitosis is the only way of how this cell can produce its progeny, because they do not form gametes and they do not form the sexual reproduction. So division of cell, which is called fission, is the only way in which these cells multiply. Regulation of cell cycle is very important because the two daughter cells that are produced should have all the normal functions. If there are any defects, genetic or functional, those cells can lead to several diseases, which is why it is important to ensure that the completion of each step is accompanied by the achievement of goals which it was supposed to have. For example, in G1 phase, cell performs its normal function. When it says that, okay, I am ready to divide, it has to enter into S phase. But from G1 to S, the cell checks if it has attained enough size to divide, if it has enough nutrients and energy to perform the cell division, if there is any damaged DNA, or what are the growth factors or the signals it has received which is prompting it to divide. Going from G2 to M, where the actual mitosis starts, this cell again checks if the DNA is damaged if the DNA replication has occurred properly, and you are ready with two sets of chromosomes which will go into two different daughter cells, and those genetic material or the sets of chromosomes are not having any kind of defects. So we have just learned about mitosis, the cell division that produces identical cells, and they happen in the non-reproductive somatic cells. In next lecture, we'll be studying about meiosis, the division that leads to the formation of gametes from diploid, that means from the organism's 46 chromosomes, you produce cells with only 36 chromosomes. So diploid cells become haploid. This is represented as 2n because it has a pair of each chromosome, and this is called haploid because it only has one copy of chromosomes. And this is how the gametes are formed. So the main takeaways from today's lecture are: mitosis ensures that there is equal distribution of chromosomes in the daughter cells, and the daughter cells receive same genetic material. This kind of cell division is required for growth, repair, and asexual reproduction in some lower organisms. Errors in mitosis can lead to several diseases such as cancer, which is why you have a tight regulation of the steps of cell cycle.